All right, great. Thank you very much. Next up, we have Dr. Elad David Amir from Astrolabe Diagnostics, and the talk title is Friendly Analysis of High Complexity Data Using Astrolabe.
Hey everyone, Tina, can you hear me and see the slides?
Yes.
Great. So first of all, thank you from MetroFlow for organizing this amazing webinar series on such a short notice. And I just checked and we have over 100 participants in here. So I would like to thank the audience as well. I know that there's quite a few opportunities for education now under the lockdown. So I am grateful that you've chosen this talk to attend. And I'm going to talk about a cytometry analysis platform that we developed here in Astrolabe. And I think that David Brooks actually gave a pretty good introduction on the motivation here, especially in the context of instruments such as the CyTOF, the Cytec Aurora, the Symphony. So just a reminder that if you have any questions, use the Q&A button below or use the chat, and I'll do my best to answer all of them at the end. So here's a conceptual way to think about analysis when it comes to flow and mass cytometry. Briefly, as the project becomes more complex, the cost of the analysis, and this means time, expertise, computational power, money, all of these go up exponentially. And I'm sure that most of the audience is intimately familiar with this area of the curve, where you have 10 to 12 markers, and you can use many of the traditional tools that exist, such as gating, such as t-tests, to analyze your data. And I think there's this expectation that as you move to higher dimensions and you get more markers, the plot is going to continue along these lines. However, based on experience with all the different high dimensional instruments that now exist, we know that the growth is actually exponential. And there's a few pitfalls that you could hit along the way. First of all, gating becomes unfeasible. You're just going to have many more gates. Moving from 10 markers to 30 markers might mean you have 70 or 80 gates to deal with. Furthermore, any change in an upstream gate, moving your CD4 versus CD8 by a few pixels, could really influence all the downstream gates. Additionally, there's many statistical pitfalls that you might have to deal with. Multiple testing correction, and similar issues that arise. So ideally, you would want to deal with these before you publish your paper, ideally as early as possible in the project. Now, many researchers acquire their data, and they're ready to analyze it, and they discover that the analysis is the greatest bottleneck for the project. And there's different tools out there. There's algorithms like t-SNE and FlowSum. There's the R programming language. And over the past few days, we've seen some excellent tools that help you access all this. Tools such as FlowJo, FCS Express, Omics, Cytobank, all of these are fantastic platforms for analyzing your data. However, all of them come with one caveat. You have to know what you're doing, and you have to know how to get there. So all of these tools are excellent Swiss Army knives if you are an expert on the bioinformatics, if you know the algorithms and the parameters. And another element to factor in here is the amount of time that analysis can take. Even if you know what you're doing, this is a case study that we've done with Mount Sinai Hospital. And here we're seeing a project that was analyzed there about 3 years ago. And the total analysis time for this project was about 20 weeks. And this factors in writing R scripts for importing data and clustering it and running t-SNE, annotating clusters, doing the statistical analysis, and sharing all of these results across the team. So just to recap, there's a bunch of amazing tools out there if you're an expert on the bioinformatics and if you have the time, the bandwidth, and the computational power to deal with the data. So what is Astrolabe? Astrolabe is a one-stop shop for analysis of flow and mass cytometry data. The idea is that we integrate into your existing experimental workflow. So you acquire your data, whether it's human or mouse or non-human primates, and your instrument can be the CyTOF, the Aurora, or the LSR2. Then the data goes on the platform, which takes care of every step of the analysis pipeline. Astrolabe cleans the data. Astrolabe can debarcode CyTOF data, identify cell subsets, conduct unsupervised clustering, and fulfill the role of the biostatistician by doing all the follow-up analysis. So if you want to identify cell subsets that differ in frequencies or markers that differ in intensity across your experimental conditions, Astrolabe does all of that. And again, the emphasis is on a turnkey solution. You don't need to know any bioinformatics. You don't need to know any programming. You don't need to set any parameters. Everything is done automatically by the platform. And then you can step in and navigate through our intuitive online interface where everything is point and click, very easy to survey the different results, different hypotheses, and decide what is interesting for your experiment. So I'm going to give you a brief demo of the platform itself. And what we're seeing here is a CyTOF dataset where the study was about chikungunya and infectious disease. And what you see here is everything you need to do to start working with the platform. First of all, you upload your FCS files. Astrolabe is cloud-based. Everything is on Amazon. And you set the experiment design. In this case, the experiment has 42 different patients. And for each one of the patients, we have 2 different time points. This table is like a spreadsheet. So if your experiment has, let's say, different tissues, if you have different treatments, different doses, different outcomes, and so on, everything can be encoded here, and it will be used for the downstream analysis. So you do this, it takes about 10 minutes, and at that point, you're done. Astrolabe kicks in, takes care of the analysis, and you can come back in a couple of hours to start navigating through the results. Our emphasis is on biologically meaningful insights. We want you to be able to think about the biology, about the immunology of the system, not about the raw data or about the parameters for different algorithms. So one of the tools that we offer is called the Cell Subset Navigator. This is a composite view of the entire experiment. So everything I'm showing you here is using data from all of your FCS files, whether you have 5 or 50 or 500. And the starting point is a list of all the different cell subsets that Astrolabe has identified in your experiment. Again, this is done entirely automatically. This is based on a peer-reviewed and published paper, so there's no proprietary algorithm here. And the process itself is very similar to traditional gating. Astrolabe first look for T cells using CD3, then for CD4 versus CD8, and so on and so forth. So already from the get-go, you can communicate with the data using the language of immunology. You don't need to annotate Wisnia maps or understand what your phenograph clusters mean. You can talk about naive CD4 T cells or about memory cells and so on. Then Astrolabe takes this complexity and organizes it using a dimensionality reduction algorithm called MDS-map. And the idea here is really to take these different subsets and put them on your computer screen in a way that you can relate to them. Each one of these bubbles is one of the cell subsets in the experiment, and their position corresponds to the similarities between them. In other words, if I color code this by CD3, you will see that all the T cells are next to each other. While if I code this by CD19 and CD20, the B cells are over here at the bottom left. So again, taking the complexity of the immune system, allowing you to think about the immunology, and presenting all of this in front of you on your computer screen. Now, immunophenotyping is just one part of what Astrolabe does. To me, the much more exciting aspect is taking the experiment design and overlaying on top of this. So I'm going to show you how we can look at the difference between the 2 time points we defined here, acute versus convalescent. And again, for your experiment, this can be anything. This can be a drug dose. This can be an outcome, responders versus non-responders. This can be conditional analysis. For example, if you want to compare males and females separately. So turning on time point, I am going to scroll down, and you will see that the MDS map has been color coded based on the fold change between the 2 conditions. In other words, the greater the fold change, the deeper the color. And just by looking at this, we can see that these monocytes over here are bright red, signifying a large fold change between the acute and convalescent time points. I can click on them and scroll down. And when I do, I will get additional information about this subset. So this section is all about intermediate monocytes. And first of all, we see a box plot with aggregate statistics. In this context, we're looking at the frequency of these cells. To the left, we see a box for the acute time point. To the right, a box for the convalescent. And we can see how the median frequency goes from 3% down to 0.5%. So a six-fold decrease. Additionally, we can see this same information on a patient-by-patient basis. So every pair of bars here is one patient. And we can see how the same trend persists across the entire experiment, with the exception of a handful of patients where the trend is actually reversed and the frequency of the convalescent time point goes up. Finally, this is all quantified using a statistical test. The FDR scores you see here is equivalent to the p-value. The higher it is, the more significant the results. To recap, Astrolabe is using your panel to identify the different cell subsets. It's using the experiment design to give you this kind of intelligence that really focuses on your research question. And finally, all these figures, all these numbers can go into your next report or into your next grant application because they are backed by the statistics. When this study was first conducted, identifying these subsets, running the analysis again, took about 20 weeks. With Astrolabe, we reproduced these results in 5 mouse clicks. Now, everything I told you so far is talking about these canonical subsets. These come from the literature. These are the T cells that you know from manual gating. To me, one of the exciting things when moving to high complexity cytometry is the ability to include additional markers that are not as straightforward as CD14 or CD20, but are still meaningful for the analysis. They have biological significance. Astrolabe offers 2 different mechanisms for incorporating these into the analysis. The first one is using unsupervised clustering. And to illustrate this point, I'm going to look at the naive CD8 T cells. When I scroll down, I see a 2-fold change in frequency for this subset. I can ask Astrolabe to cluster these cells by switching to the profiling level. So the bubbles you see here, these are canonical subsets. They come from the literature. When I switch to profiling, you will see that I have many more bubbles. I have many more subsets. These are still the canonical subsets you're familiar with. These are still naive CD8 T cells. These are still memory B cells. However, they've been broken down using unsupervised clustering. This step is completely agnostic. You don't need to choose any parameters. Astrolabe does it for you. And the platform also offers this volcano plot visualization to help you navigate through this wealth of information. So in this plot, every dot is one of the clusters. The higher they are, the more significant the trend for this cluster between the 2 time points. And the platform has already highlighted the naive CD8 T cell clusters. So we can see 1 and 3 more here at the bottom. So one of these clusters is much more significant than the others. We can focus just on this cluster and scroll down, and we will see a 10-fold change in frequency between the 2 time points. Just to recap, when I go back to the canonical assignment, I see a 2-fold change. And then when I look at the clustering, a much more significant 10-fold change. Sorry about that. Here it is now. Now, I mentioned this paper has been published a while ago. The manuscript doesn't mention CXCR3 at all. And in fact, when I reached out to the authors of the paper, they weren't aware of this trend. They used traditional gating to reproduce this. And they confirmed that there is a CXCR3 positive compartment, which is almost exclusive to the convalescent time point. And to me, this really highlights the benefit of Astrolabe. Yes, there's the element of time, but more importantly, it allows you to squeeze the most out of your data. As you're moving into high complexity cytometry, or if you're already using the CyTOF or the Cytec Aurora or the Symphony, you know that these experiments are tricky. You know that they're expensive, they're time consuming, and more often than not, they use precious samples. You want to get the most out of these data sets. You want to be able to feel confident that you've explored the different hypotheses and the different trends, and Astrolabe really liberates you to do that. So I mentioned that we have 2 different ways to look at the markers in the experiment, and clustering is one of them. The other way to do so is using a module called differential expression analysis. And this is especially relevant for activation markers, for exhaustion markers, cytokines, chemokine receptors, and so on. And just to highlight some of the Astrolabe results for this data set, I'm first going to show you PD1. And in these visualizations, every panel is one of the cell subsets we're looking at all of the samples in the acute time point and the convalescent time point, and we're assessing the level of PD-1 across these samples. So think about it, we are integrating a lot of information to generate these figures. Everything is quantified, everything has p-values and fold changes. And just by eyeballing this, we can already see some interesting trends. For example, in the myeloid compartment, the level of PD-1 seems to go up between the two time points, while in the T-cell compartment, the level of PD-1 seems to go down. And one of the things that we know is that during the acute time point, the response goes to the immune system, to the innate immune system. During the convalescent time point, the response comes from the adaptive immune system. We can see this in cell subset frequencies, but we can also see this in the expression of PD-1 across these different compartments. We can also reproduce what we saw with CXCR3. So when looking at these figures to your left, you can see the naive CD8 T cells. And once again, we see the increase in CXCR3 expression in these cells between the convalescent and the acute. However, we do not see a similar response for the naive CD4 T cells. When we inspect the T cells as a whole, we see that as a compartment, they seem to have this trend. However, the naive CD4 T cells are actually the exception here. So we've gone through a bit of a journey where we were suspicious about the naive CD8 T cells and CXCR3, and by using an orthogonal analysis, we can actually see that this is a T cell wide trend, and the naive CD4 T cells are the interesting bit here. And finally, one last example for the differential expression analysis. This panel includes CHIKV, which is a marker for virus infection. In other words, it's on the surface of those cells that are infected by the chikungunya virus. And looking across the different immune compartments, we see that it doesn't change that much between the two time points, except for the myeloid compartment. And if I recall correctly, one of the hypotheses from the paper is that the chikungunya virus targets the myeloid cells, and this is reproduced very nicely by this kind of analysis that Astrolabe provides. So just a brief reminder that if you have any questions, now would be an excellent time to ask them. Just use the Q&A button at the bottom. And I want to go back to this slide I've shown you before, where analyzing this data set took about 20 weeks of time about 3 years ago. Reproducing this study with Astrolabe should take you somewhere between, let's say, 12 hours and a day. And a bench scientist who is working on her laptop computer can upload the data, can set up the experiments in 10 hours, and within an hour or two, come back and navigate through all of the results. And then you can focus your attention on what matters, not about parameters or algorithms, but about canonical subsets, about the actual clusters, about the trends that you want to see in your experiment. And just to clarify, Astrolabe is not going to solve all of your problems, but it is going to help you with this burden of analysis. And at this point, we've seen multiple cases where researchers have been sitting on a project for months, within a few days of using Astrolabe, reached a publishable manuscript. It's really about lowering this burden, making your life easier. So with that, I'm going to thank MeasureFlow again, and I'm going to thank you, the audience, for attending, and I'm happy to take any questions.
Thank you so much, Elad. That was absolutely wonderful. And we do have a few questions that have come through, so we'll get started. So Thornston asked, does the software have 21 Part 11 CRF compliance features that it can be used in a regulated environment, or is it currently for research use only?
At this point, we're for research use only. However, we are engaging with a third-party firm that is taking us through Part 11 compliance. And I hope that we will be Part 11 compliant within the next few months.
Perfect. Then we also have a question from Rohit, and they're asking, is it possible to analyze without prior knowledge-based results, i.e. CD14 expression in T cell subsets in some patients, just like citrus?
Yes, and that's an excellent question. I think that every experiment is unique. And although it's nice to have the reference and to know what you expect, there's always exceptions. There's different ways that Astrolabe helps you with that. One of them, and let me go back to the platform, one of them is if you know that there is a certain subset you're expecting and Astrolabe does not find it, we can work with you to update the platform and add it. At the end of the day, it's very simple rules. Again, very similar to traditional gating. For example, if you know you're looking for macrophages and Astrolabe doesn't find these by default, we can add the appropriate rule here. Then every cell that doesn't fit into the expected compartments is going to be part of the analysis, and Astrolabe is going to label it appropriately. For example, we have this bucket, this subset called T cell unassigned. And these are cells that are T cells. They're CD3 positive. However, we could not fit them into the memory versus naive, into the CD4 versus CD8 separation. It's here, it's part of the analysis, it's going to be clustered, and you as a researcher can step in and decide whether it's meaningful to your experiment or whether it's just noise or junk. Using these cells as an example and scrolling down, their frequency is very low, below 0.1% for most of the samples. And upon closer examination, we're pretty much convinced that they just slip through the cracks. Even when you draw a traditional gate, there's always some cells that don't fall into any of the gates. This is probably what we're seeing here. However, if they were interesting, for example, if they were significantly expressed by the different conditions, we would see it here and we would probably dig a bit deeper. So again, the emphasis is on allowing you to survey the data and make the decisions in as much an informed fashion as possible.
Perfect. So I personally have a question. I saw recently that it seems that there's a Flojo plugin for Astrolabe, and that's not something I've seen at this point. So I'm a bit curious as to what that plugin does in order to be able to bring in an Astrolabe analysis into that other third-party software as well, you know, for people that are familiar with it.
Thank you for asking that. And I started this talk by mentioning some of the excellent other tools that exist out there, like Flojo, Omics, Cytobank, FCS Express, etc. And Astrolabe was designed to complement them. In other words, we're not looking to take your Flojo. I know that some researchers are a bit worried about that. Vice versa, we work with Flojo and the other platforms to help you take the Astrolabe results into these other pieces of software. And in the context of the Flojo plugin, what it allows you to do is to download these FCS files up here. And these are almost identical to your starting FCS files, except they have additional columns with this information in them. In other words, you can now go into Flojo, push a button for the plugin, and it will automatically show you all of these different compartments. Now, if you want to do a follow-up analysis, if you want to present a heat map, if you want to run t-SNE, if you want to run t-SNE but only on the naive T cells, you can use this information to greatly facilitate your downstream analysis. And in this context, I often present Astrolabe as a staging ground. It's not aimed to solve all of your problems. Sometimes it can, when depending on the design, but more often than not, you're going to have follow-up questions, you're going to have follow-up analyses. And our goal is, again, to serve as the staging ground for these analyses. Instead of starting from the raw FCS data, you can start from the Astrolabe result. You can make much more informed decisions on the kind of tools you use and the kind of questions you ask.
Perfect, thank you. And then one more question. So does the platform handle incredibly rare cell types as well? So if you had, say, if you collected 1 million events and there was an incredibly rare cell, like a 1 in a million type of cell, how does it handle that? And are you able to pull out populations like that?
Well, I think that 1 in a million out of 1 million, probably not, just because we can't quantify it very well. Looking at existing examples, we identify plasmatidoid dendritic cells and basophils on a regular basis. And for many samples, these can be less than 0.1%. Likewise, we had experiments where a researcher was keen on finding a population of, let's say, 15 to 20 cells, and Astrolabe was successful in finding them. With that said, your mileage may vary. One of the things that we do is we offer a free trial. So if you do want to test the platform using any of your data, then by all means, please send me an email. And we can take your data, run it through the platform, see if it answers the specific questions that you have in mind.
Okay, perfect. Thank you so much, Ella. As usual, absolutely wonderful talk.
Thank
And
you.
those are all the questions we have. So just wanted to say thank you again for a wonderful presentation.
Thank you, Kathy. Stay safe, everyone.